Hey folks, welcome back. All right, if you're starting to get a little worn down with your macromolecule videos, just note this one is relatively short. Nucleic acids is pretty quick. So, nucleic acids, there are two famous ones, right? DNA and RNA. DNA sort of gets the praise and recognition because it's this important molecule of heredity. RNA doesn't get as much attention, right? It's like the offensive lineman of the cell. It doesn't get as much recognition for its importance, but RNA is really important because it actually does the work of building these molecules and a key role in making proteins. But the thing that I think is sort of helpful to consider is proteins do the work of the body, right? Again, proteins, the little machines that carry out the reactions that make life happen. But DNA is important because DNA is the instructions for making the machines. So DNA has the instructions for how to build all these important machines. RNA does the work of actually doing the building. And so these molecules make it possible to construct and run cells and then pass that information on to future generations of cells. So these are incredibly important molecules for maintaining and operating life. Now, they come in two varieties. Again, there's DNA, which is a deoxyribonucleic acid, and RNA, which is a ribonucleic acid. They differ slightly in their sugars, and they differ just a little bit in which combination of bases they have. But again, these molecules work together to build and instruct the construction of proteins. Let's see. Now, it is a true macromolecule. It is a polymer. The polymer is sort of a funky name. It's called a polynucleotide. The monomer is what I'd like you to focus on. It is a nucleotide. So the monomers of carbs were monosaccharides, right? Saccharine means sugar. So monosaccharides are single sugars. Nucleotides are the monomers that build up DNA and RNA. Now, please pay particular attention to the structure of a nucleotide. We've discussed the atoms that make up carbs and lipids. Carbs are CHO with a lot of O. They dissolve in water. Lipids are CHO with very little O, so they tend to be nonpolar. Now let's look at the nucleotides that make up DNA and RNA. You'll notice you have a phosphate group, all those oxygens there. There's a sugar, a bunch of oxygen. Nitrogenous bases are nitrogenous because they have a bunch of nitrogens, which are also pretty electronegative. So the way I remember the structure of nucleic acids or nucleotides is to think of CHOMP. They have carbon, hydrogen, and oxygen, but they also have phosphorus and nitrogen. So carbon, hydrogen, oxygen, and then nitrogen and phosphorus. So CHOMP. If you see phosphorus, that's a giveaway that you're looking at a nucleic acid, right? Let's see. So you have the phosphate group, the sugar, and then the nitrogenous bases are where the codes are stored, right? So the order of the bases sort of dictate the codes in DNA and RNA. Now, these nitrogenous rings come in two varieties. They're single rings or double rings. The purines have two rings. The way I remember the purines are A and G. I think of Purina dog chow as an agricultural product. I mean, I don't know how relevant that is to you guys, but I just remember Purina dog chow is an ag product. So A and G are purines, are double ringed, and then pyrimidines are single ringed. And so, a double ringed nitrogenous base pairs with a single ring nitrogenous base. That recognition was the key breakthrough for Watson and Crick for figuring out the structure of DNA. But again, that's not super important right now. Now, one thing I would like to do is draw your attention to the structure of DNA. DNA is double-stranded, two strands of DNA. They're held together by hydrogen bonds between the nitrogenous bases, A pairing with T, C with G, again, purine with pyrimidine. But the strands are anti-parallel. What that means is they run in opposite directions. So how do they run in certain directions? Well, remember back when we were numbering carbons, you have your asymmetric carbon, that's carbon 1, and 2, 3, 4, 5. If you look at carbon 5 here, what partner does it have? It's a phosphate group. If you look at carbon number 3, so there's 1, 2, 3, carbon 3 has a hydroxyl group. So the strands have what's called a 5' end and a 3' end. And if you'll notice between neighboring molecules, the 5' ends and 3' ends are on opposite sides. So they run in different directions. Again, that's not super important now, but next semester we'll explain why having strands that are anti-parallel like this means that we almost die. So again, don't sweat it for now, but we'll talk about that next semester. Okay, that's it on lipids. Again, it's pretty quick. We got one segment remaining, looking at proteins. So you're getting close. Stick with it.